Good morning, my fellow scientists. It is Thursday, June 22nd, 2017. I want to talk a little more about aptamer-based therapeutics. I talked yesterday about the idea of an alphamer, or an alpha-galactose-tagged aptamer that could be used to bind to a pathogenic bacterium and then flag that bacterium for removal by the immune system. Really cool approach. The company that makes the alphamer, called Altimmune, was bought out this year by Centauri Therapeutics out of the UK. I hope that they are able to develop it quickly into something that can be tried out in clinical trials. So that's exciting. I read about another technology. A company called Oisin Biotechnologies develops senolytic drugs that are supposed to target and destroy senescent cells, essentially alive but not propagating properly anymore. These cells are thought to accumulate with age. So they're trying to develop this method for killing senescent cells. But senescent cells, kind of like cancer cells, are human cells, and as a consequence, you have to be really careful how you go about killing them. If you are too aggressive, you start killing normal tissue. If you're not aggressive enough, it leads to resistance. So how do you do that? How do you tune whatever approach you're taking? The approach that the engineered senescence group took was to introduce a suicide gene that's going to kill the cells that it is expressed in, but then induce that suicide gene at a very precise dosage using a drug that enables the protein of the suicide gene. What that means is that you can tune the amount of suicide gene activity in the body by tweaking just a small molecule drug dose, which is considerably easier and safer than a large molecule or gene that may be harder to regulate. Makes the whole thing a lot safer. And I was thinking to myself, could we add that same kind of complementary safety protocol to an aptamer-style therapeutic? And I think the answer is yes. I think that the answer is to go to the strategy by Sullenger, where you introduce a complementary antidote if things get ugly. So in that case, the Zollinger system used an aptamer against a protein called thrombin, which is involved in coagulation. When the aptamer is introduced, it inhibits coagulation, can help prevent strokes, but if too much of the aptamer is present, it can lead to uncontrolled bleeding, which is obviously bad. So there needs to be some way to provide an antidote to that aptamer in order to shut it down and restore enough clotting activity to prevent bleeding. And that actually can be another piece of DNA. Aptamers are one piece of DNA. If you introduce another piece of DNA that binds to that DNA, it takes on a double helix structure we're all familiar with. That's not an aptamer anymore, and the whole thing shuts down. So could you do that with the Altimmune? I suspect so. I suspect you could have the alphamer bind to bacterial cells, and if you start to see off-target effects, you could introduce a complementary oligonucleotide that would disrupt the alphamer and shut it down. All of these technologies are still speculative. They're not a cure for cancer yet, but there's some hope. This kind of thing could be a step toward, whether it's cancer or multi-drug resistant bacteria or just aging, that these kinds of technologies could eventually build up an arsenal of tools to help prevent these. And I find that pretty exciting. So if you like hearing about this kind of thing, whether it's batteries or biotechnology, I hope you'll tune in tomorrow. We update Monday through Friday. We talk about science and chemistry and biochemistry here in the Allen Lab.